Here we go. Thank you very much for the introduction. In everyday life, we have to take decisions based on our preferences, so-called value-based decisions. A prominent example of such decisions are food choices. For example, tonight you will have to decide if you want to go to a food stall and try a Singaporean dish, or you want to go to a more fancy restaurant. However, please remember that the food quality is the same, since, as you know, in Singapore, even the food stalls can have 1 Michelin stars. In my talk, I aim to reveal the causal brain mechanisms that underlie such value-based choices by combining online TACS with functional imaging. In all the experiments that I'm going to present today, we use the same behavioral task, where in every single trial, the participants had to decide between the two food items displayed, the item that they would like to eat most. We selected pairs of food items based on previous ratings, and we choose 4 evidence levels, where the evidence is defined as the difference between the subjective ratings of the two displayed food items. And as a behavioral index, we measure the choice consistency, defined as the probability that the subject will choose the food item that they rated higher at the beginning of the experiment. And previous research suggests that there is a direct link between the choice evidence and choice consistency. For example, if you have to choose between two food items that you like similarly, the choice consistency is low, the task difficulty is high, and the evidence is also low. In a first experiment conducted in our lab, my colleagues demonstrate that the choice consistency correlates with the strength of the coherence in mid-gamma frequency between frontal and parietal areas. And this was specific for value-based choices, and this effect was not seen during our control task, perceptual decision task, where we used the same set of stimuli. Consequently, we wanted to test if this frontal parietal coherence is causally relevant for value-based choices. We did that by applying the 3-electrode TACS setup, which is thought to decrease the coupling between the two stimulated areas by applying antiphase TACS oscillatory currents, 180 degrees between the two currents over the two stimulated areas. In the current experiment, we placed 2 main electrodes over the frontal and parietal cortices and the reference electrode over the shoulder. And indeed, what we observed is that compared to sham stimulation, antiphase TACS decreases the choice consistency. This was specific for value-based choices and was not seen in our control task, perceptual decision task. However, the neural mechanisms that underlie such behavioral effects are unknown. Thus, the aim of the main experiment that I'm going to show you today is to reveal these mechanisms by combining online TACS with fMRI. And what are the possible mechanisms? Previous literature suggests that there are at least 2 possible mechanisms. On one hand, previous research suggested that value is constructed in the frontal cortex and the information is transferred to the parietal cortex through direct communication for the implementation of the motor choice. If this is the case, then we expect that antiphase TACS will disrupt the functional connectivity between the stimulated frontal and parietal areas. On the other hand, other research suggests that value is constructed in both parietal and frontal areas through direct communications with the subcortical regions within an extended valuation network. This network contains regions that exhibit high activity with the reward, for example, ventral striatum or VMPFC, as well as memory networks that exhibit, that encode memory samples of previous experiences with the food stimuli, such as hippocampus. If this hypothesis is true, then we expect that antiphase will disrupt the value representation in these subcortical valuation regions. Furthermore, we expect that antiphase TACS will disrupt the connectivity between these subcortical valuation regions and the two stimulated frontal and parietal areas. In order to test this, we combine online TACS with fMRI by means of an already established technical setup. We implied the within subject design where block of 3 minutes of TACS stimulation were alternated with sham stimulation, while the participants perform exactly the same task where they had to choose the item that they would like to eat most. First, we wanted to investigate if we are able to replicate our previous behavioral results. And indeed, what we observed is that also during the current TACS fMRI study, anti-phase TACS compared to sham decreased the choice consistency. And this shows one more time that the frontal parietal coupling coherence is relevant for value-based choices. Next, we tested our first hypothesis that stated that anti-phase TACS will disrupt the direct communication between frontal and parietal areas. However, we did not see such an effect. We did not see any impact of the stimulation on the functional connectivity between frontal and parietal areas. We then tested our second hypothesis that stated that anti-phase TACS will disrupt value construction. And indeed, what we observed is that anti-phase TACS decreases the value representation in two subcortical valuation regions, namely hippocampus and ventral striatum. The Ball signal in these regions highly correlate with value representation and during sham condition, and this is disturbed when anti-phase TACS is applied. This is a main effect, overall effect, meaning that anti-phase stimulation affects the value representation similarly in all subjects, irrespective of the impact of the stimulation on choice consistency. We further investigated the link between these two subcortical valuation regions, and we saw that there is a high correlation between the impact of the stimulation on value representation in hippocampus and ventral striatum. This suggests that this is sort of consistent with the notion that these areas are both involved in value constructions for choice. To further consolidate our first hypothesis that anti-phase TACS disrupts value constructions, we conducted two PPI connectivity analysis. In the first one, we said the seed region in hippocampus. We did not see main effect of the stimulation. However, we saw an interaction between the impact of the stimulation on choice consistency, on behavior, and the impact of the stimulation on the value-related functional connectivity between the seed in hippocampus and the stimulated parietal cortices. More precise, the stronger the decrease in the choice consistency, the stronger the decrease in value-related functional coupling between hippocampus and stimulated parietal cortex. Similarly, a second PPI analysis with the seed in ventral striatum revealed that the stronger the TACS decrease on choice consistency, the stronger the decrease in value-related functional coupling between ventral striatum and stimulated frontal cortex. Thus, these two connectivity analyses revealed that anti-phase TACS decreases the functional connectivity between frontal and parietal areas and subcortical valuation regions in a manner that is dependent of impact of the stimulation on behavior. To conclude, we show a brain. We reveal the causal brain mechanisms that is relevant for consistent value-based choices. We implied TACS protocol, anti-phase TACS, that was meant to disrupt the frontal parietal coherence. Anti-phase TACS decreased the choice consistency as compared to sham. However, this behavioral effect is not reflected in disruption of the direct communication between frontal and parietal areas. Instead, we see that anti-phase TACS disrupts the value representation in two subcortical regions within the valuation network, namely hippocampus and ventral striatum. Furthermore, we show that anti-phase TACS disrupts the connectivity between the two stimulated areas, frontal and parietal regions, and these subcortical valuation regions, in a manner that relates to the change in behavior due to the stimulation. Thus, our results suggest that the coherence between frontal and parietal regions seen in EEG and targeted with TACS is not reflected in a direct communication between frontal and parietal areas, but rather these two areas functionally connect with subcortical areas within valuation network in the service of constructing choice for value-based choices. More general, we show that combining TSS with fMRI online can reveal crucial information on the interplay between brain mechanisms and behavior. I would like to thank my collaborators and to you for your attention. Thank you very much.